Have almost 161 participants right now. So we'll start with the session. The session fourth of the day is being moderated by Dr. Dilafrose. Dr. Dil is professor and head in the Advanced Center for Immune Genetics, Sher Kashmir Institute of Medical Sciences, JNK, India. She is the Vice President North for Indian Immunology Society. Her research interests include immunogenetics, immunopathogenesis of cancer. She is the visiting professor to Division of Oncology, University of Linköping in Sweden, and University of Louisville School of Medicine in US. She has guided several basic and clinical graduates. May I hand over the stage to Dr. Dil for the moderation, please?
Uh, Thank you, Gulmeher. Can you all see me? I think I'm audible and can see me as well. Should I go ahead? So, what a stimulating day it has been, and start by thanking Dr. Rora and Dr. Amit Avasthi for this wonderful FIMSA virtual immunology course from basic to advanced immunology. And without much, this is the penultimate session of the day, and after this, there is going to be the keynote session. So I start with the session 4. We have two speakers in the session. Prafulla Kumar Taylor, Dr. Prafulla Kumar Taylor will be talking about the pattern recognition receptors. That will be followed by Dr. Monadipa Ghosh. She'll be talking about inflammation and the role of neutrophils in innate immune response. It's my great privilege to introduce Dr. Prafulla Kumar Taylor, who is senior scientist at the National Institute of Immunology, New Delhi, India. Dr. Taylor worked for his PhD under the guidance of Dr. Robin Mukhopadhyay from Tata Memorial Center, Mumbai, where he went on to successfully work on the interferon therapy for CML patients, and hence his keen interest in the interferon regulatory factors. Then he went on to work with Dr. Keiko Ozato at National Institute of Health on regulation of type 1 interferon induction in dendritic cells by viruses and TLRs, that's toll-like receptor ligands, and the innate immunity. uh, Dr. Prafulla Taylor is a recipient of the Ramalinga Swamy Fellowship from the Department of Biotechnology and recently a Mid-Career Scientist Award from Indian Immunology Society. He has been working on the functional specificity of diverse DC subsets, and his lab at National Institute of Immunology has contributed towards understanding the development of classical DC subtypes and focuses mainly on understanding the mechanisms of DC diversity development. So today he's going to talk about the pattern recognition receptors, which are key players in the innate immunity. Uh, do we have Dr. Prafulla Taylor on the? Is he there, Dr.
Hello, can you
Prafulla
hear
Kumar?
me?
Yeah,
Hello?
yeah,
Yeah,
yeah.
okay, I'm here. I'll start sharing the screen. Mm, Okay, so first of all, thank you very much for a kind introduction, and thanks to the organizer for giving me opportunity to present pattern recognition receptors here. So as I mentioned that my laboratory, as mentioned, my laboratory mainly works on dendritic cell development from a progenitor, and that too mainly transcriptional regulation of diverse DC subtypes. And why I'm interested in DC subtypes, it is as shown here. There are different DC subtypes which each of them have a specific functions, and that makes it more interesting. How come from a single DC progenitor cells, a different DC subtypes have developed which have a functional diversity. Okay, and they play a very important role in TLR response. But I'm not going to talk about this work because I was under impression that this is going to be a teaching symposium or a sort of refresher course kind of thing. So I selected from the available courses to talk about pattern recognition receptor. And that is what I'm going to talk about today. My work was made much easier by Dr. Himanshu Kumar and Dr. Osama Takeuchi, who introduced almost everything that I'm going to talk about today. And as I'm not going to present data today, uh, as I'm not going to make any data presentation, it's going to be more of an introduction to pattern recognition receptor. So most of the panelists might find it boring, and if required, some of you can take some break. This is going to be mainly talking about pattern recognition receptors. Okay, so going ahead, as Dr. Himanshu Kumar also presented, and it, as we all know that we have microbes all around us, okay, and we are surrounded by microbes, and we live with microbes, and we are constantly exposed to pathogens or say microbes which can cause diseases, and hence we need to protect ourselves, okay, and for that evolutionarily our body has developed innate immune response as well as adaptive immune responses. Okay, now going to pattern recognition receptors that I'm going to talk about, the word was first coined in 1989 by Dr. Charles Janway. Okay, at that point of time, nobody knew about Tlrs. Nobody knew whether there is a pattern recognition receptor will be there or not. But he proposed in 1989 that there will be a non-clonal pattern recognition receptor which can recognize pathogen as they are, and that it is different than the clonal receptors like T cell receptors and B cell receptors, right? Antibodies that recognize the specific antigen. Okay, but the major impetus came when, and also Dr. Charles Janway had identified that this pattern recognition receptor signaling will play a very important role in immune system. In 1997 itself, he had predicted that the signaling through pattern recognition receptors can play important role in activating T cells, in defining B cells activation, and also macrophage activation. So he was a visionary in his own sense that we all know, but all this what we know today about pattern recognition receptors was already predicted or has been put forth by Dr. Charles Janway at that point of time itself. Okay. Now, going to now, since we come to pattern recognition receptor, what is pattern? Okay. Generally, I enjoy talking about this subject when there is an interactive session, but it's a virtual. So let's go ahead. And what is pattern here? So if you take a dictionary meaning of pattern, it says an arrangement of repeated parts or a decorative design. Okay, so something that is repeated and a regular recognizable way that something is done, which means that something has to be repeated and that should have been recognizable way, and that is when it is called a pattern. Okay, and this is the pattern that our immune system tries to identify, and that's why it is called pattern recognition receptors. PAM. What are the PAMs? Even PAM word was coined by Dr. Charles Janway in 1989 itself, where he had mentioned that pathogen associated molecular pattern is the one that will be detected by the pattern recognition receptors. Now, what could be different PAMs? Like if you look at bacteria and viruses, there are different infectious agents like bacteria, viruses, fungi, and they all have different cell wall components, or they have different nucleic acid material. And those are PAMs which are different than what our human body cells are. And that is what is recognized as non-self, and that is what a pattern recognition receptor detects and gives response to. And as it was mentioned earlier that innate immune system was considered very crude in earlier stages of studies, and hence that there was a dogma that stayed till today that innate immune system is not specific, and that is what pattern recognition receptor will change. So after once I finish my talk, we may be able to appreciate how specific innate immune responses are. Okay, so let's go on. So there are different type of pattern recognition receptors, and that can be simply classified into toll membrane bound receptors and a cytoplasmic receptors. And membrane bound receptors are like toll-like receptors, mannose receptors, or dectins, and a cytoplasmic receptors are RNA helicases, node-like receptors, or DNA sensing receptors. So this is very simple classification. Now let's go on to the history of TIR domain, because TIR domain is the one that defines toll-like receptors. Okay, so in 1988, IL-1 receptor was cloned, and that receptor had a cytoplasmic domain that could not be matched with any previously known cytoplasmic domains. Okay, and IL-1 receptor played a very important role in T cell activation, cartilage breakdown, and activating acute phase protein response, and that became a very intense focus to study IL-1 receptor. And that is what I'm once again showing here, that 1989 itself, Dr. Charles Janway had proposed concept of PRRs. In 1991, while studying the sequence of toll in Drosophila, they identified the cytoplasmic domain of IL-1 receptor matched somehow or with that of toll receptor in Drosophila. But at that point of time, the toll receptor in Drosophila was linked with promoting development of dorsoventral polarity of embryo. Okay, so it was important for developmental biology. And here we had one receptor which was important for immune responses. Now, in 1994, the study showed that plant-derived, host-derived N protein provides a resistance to tobacco mosaic virus. So here it is a plant protein which provided responsible for the resistance to tobacco mosaic virus. And this plant protein, N protein, also had this TIR domain. Okay, so there is three protein which we known to have which was found to have this TIR domain, and hence it is known this domain is named TIR because it is found in toll, IL-1 receptor and resistance, that is TIR domain. Okay, later on it was shown that even in Drosophila, this TIR toll receptor was responsible for antifungal response, and then TLR4 was identified and many where TLRs were identified. But much before the TLR identification or patent recognition receptor identification, Charles Janvey had already proposed that it will play a very important role in our innate immune system. Now, here, this is a cytoplasmic domain that I'm talking of, TIR domain, which is conserved or which is similar in IR1 receptor as well as toll-like receptors. So now toll-like receptor has intracellular, that is TIR domain, cytoplasmic, which is TIR domain, and extracellular domain, which is called LRR domain. That means leucine-rich repeats. And these leucine-rich repeats, based on their arrangement, defines which ligand it is going to detect and which ligand it's going to recognize and send a signal downstream. So here are different TLRs that I'm showing with their ligands. Like you can see here, TLR9 recognizes CpG DNA. Our mammalian DNA is methylated, whereas bacterial DNA or viral DNA is not methylated, and hence it can be detected by CpG DNA can be detected by TLR9. Even TLR8 is single-stranded RNA. TLR7 is also single-stranded RNA. TLR5 detects flagellin. TLR4 detects LPS and many other factors as listed here. TLR3 recognizes double-stranded RNA, and these three TLRs combines with each other and recognizes various proteins or molecules from bacteria and yeast. Now, why this is so simplified, pattern recognition receptor is very simplified view towards a self versus non-self, because here you don't look at you don't look at heptin, or you don't look at an antigen from a very motif in terms of responding to antibody or specificity, but here is broad classification. Like here you can see bacteria, these TLRs can recognize bacteria, viruses here, and protozoa or fungi. So if it is a single-stranded RNA virus, then TLR7 or 8 can recognize it. Or it is a double-stranded RNA virus, TLR3 can recognize it. Or if it's a viral DNA or a bacterial DNA which is not methylated, TLR9 will recognize it. So it is simply self versus non-self. That means that our immune system is not interested in details, but it is whether you just want to identify self versus non-self and respond to it so that we can get rid of these pathogens from our body. Okay. Now, based on, as I mentioned, that some of the TLRs detect nucleic acids, okay, and some of most of them are detecting cell membrane bound molecules or the components of a cell wall, and that is why the distribution of TLRs are in such a way that the TLRs which recognize the membrane structures are expressed on the cell membrane, and the TLRs which recognizes the nucleic acid are expressed in endosome, which is where the most of the bacteria are going to get engulfed, and that is where the bacterial nucleic acid or viral nucleic acid is going to be coming in contact with TLR. So that is how evolutionary nature has distributed TLR in a way that it detects its ligand. Now, coming to the TLR, see whatever is happening on the cell membrane, it ultimately has to send a signal to nucleic acid and at the nucleus, and that is where the transcription is going to happen, from where some of the molecules are going to get synthesized, and it will be secreted out for other immune systems to work on or get activated on. Okay, so let us look at the signal transduction event of TLRs. Okay. So first that comes in signal transduction event is MYD88. MYD88 was identified while working on IL-6 induced myeloid differentiation. Okay, and MYD88 has a TIR domain. Okay, suggesting that this TIR domain will interact with TIR domain of TLRs and can assess anchor there and send signal downstream. Now, MYD88 also has a death domain, which is also found in TNF alpha receptor or TREF molecules, and TNF alpha receptor, and this death domain is responsible for inducing death of the cell. So MYD88 has TIR domain and as well as death domain. And most of the TLRs use MYD88 as an adapter molecule, like TLR2. Here you can see TLR2, TLR5, TLR6, okay, TLR10, 11, and 7, 8, and 9. Next in queue is a IRAC, that is IL-1 receptor associated kinase. Now IRAC1 or IRAC has DD domain. So that's how it can interact with MYD88. Now we have seen TLR have TIR domain, MYD88 has TIR domain, so there is an interaction. Now MYD88 has DD domain, sorry, death domain, and similarly IRAC have death domain. So they both can interact with each other through this death domain and can send signal downstream. So activation of IRACs is through phosphorylation of this molecules, IRAC molecule. Now, once the IRAC gets phosphorylated, it interacts with a TRAF molecule, which is TNF alpha receptor associated 6 molecule. Or there are 6 members of TRAFs. Most common interacting or most commonly studied is TRAF6, but TRAF3 also has been shown to be very important molecule in this TLR signaling cascade. Okay, the TRAFs directly interact with, as I said, it can directly interact with the TNF alpha receptor and indirectly to TLRs through IRAC molecules here. Now, moment TRAF interacts with IRAC, it gets dissociates from it, and then this complex goes and associates with TAK, which is TGF beta associated kinase 1, TAC1 with TAB molecules, and here it comes in contact with the ubiquitination machinery. This ubiquitination machinery ubiquitinates TRAF6 and in result ubiquitinates TAB molecules. This exposes TAC1, which autophosphorylates itself, and TAC1 gets activated. Now, activated TAC1 goes and phosphorylates IKK complex. Now, what is IKK complex? IKK complex is I kappa kinase. Okay, so its role is to phosphorylate I kappa B. Now, this I kappa B, when it gets phosphorylated, it gets degraded, and it is inhibitory component of NF kappa B complex. Okay, once this phosphorylation degrades I kappa B, then P50 and P65 complex is released, which enters to the nucleus and goes and binds to NF kappa B binding motif in most of the cytokines which are inflammatory in nature. And once this is made, these cytokines are secreted outside the cell, and then the immune system gets activated. This is how the signal is transmitted from TLRs to the nucleus. But now, is there MYD88 independent signaling mechanism? Because we all know immune system is so diverse that it does not rely only on one pathway or one molecule to do its job. Otherwise, everything will collapse, right? So then when they were looking at the studying TLR MYD88 knockout cells, they activated TLR4 through LPS stimulation. They found that there was no inflammatory cytokines that are made like TNF or IL-6 or IL-1 beta, but they could detect interferon beta or interferon inducible genes, which is a bit later than that of what MYD88 positive cells or MYD88 sufficient cell would have shown, suggesting that there is a MYD88 independent mechanism, and there is some molecule, adapter molecule, which is not, which is different than MYD88. And the search led to identification. Okay. Before search led to identification of molecule named TRIF, which is a TIR domain containing adapter inducing if interferons, because that is what was induced in MYD88 knockout cells. Okay, and this TRIF, when they overexpressed TRIF, they could see the induction of NF kappa B promoter as well as interferon beta promoter, and which could not be blocked by MYD88 or TIRAP, which I'm going to just talk in few minutes or next slide. And later on, it was found that TIR domain containing adapter molecule TREM was identified as an adapter for TRIF molecule, which comes before TRIF, and that interacts with the TLR. And TLR3 is the one that exclusively transduces signals through TRIF, whereas TLR4 can send signals to MYD88 as well as TRIF. Now, when people were looking at the identification of embodied independent adapter molecule, there were two molecules identified at the same time by two independent groups. The Ruslan-Mazditov's group identified TIRAP, which is known as TIR domain containing adapter protein, okay, and Luke O'Neill's group identified embodied adapter-like protein, which is MAL, okay. But later on, it was identified that these two molecules, both the molecules are same, and they are not embodied independent mechanism, but they are the one that anchors embodied to the TLRs. So there are these two molecules are not independent mechanism, but are very important for TLR signaling. Okay. So now till now we have seen that how the signal transduction works. And here you can see that TRIF molecule can directly activate NF-kappa B or IKK complex. And then you can see IRF molecules are getting activated, the molecules that I work predominantly on. And this IRFs then goes to the nucleus and binds to the promoters of interferon or type 1 interferon promoters and produces type 1 interferon that plays very important role in antiviral responses. Now, so till now we have seen MYD88 as well as TRIF. Here is a TRIF, which is adapter molecule for TLR4 as well as TLR3, which recognizes double-stranded RNA. Now, if the TLR signaling keeps continuously activated or it is activated, it might lead to hyperimmune activation or can lead to autoimmune reaction. And that's why we need to control the TLR signaling. And as Dr. Takeuchi showed just recently, there are many molecules that play very important role in controlling the TLR response. IRAC-M, which does not get phosphorylated, can also go and bind here and attenuate the signaling through TLR responses. SOX1 that was mentioned by Dr. Takeuchi, MYD88 splice variant, which does not have DD, so it cannot transit signal downstream. Okay, and recently many deubiquitinating enzymes have been identified, which play a very important role in abrogating the TLR signaling. And one of these molecules was, even viruses utilize these deubiquitinating enzymes. And one of such enzyme was identified and described by Dr. Himanshu Kumar recently, where he showed that HCMV deubiquitinase enzyme can deubiquitinylate many TLR component or TNR machinery and attenuate the signaling here. Okay, so that was about the TLR signaling. Okay, now this is there are many other TLRs have been recently identified which recognizes ligand, and one of that such is TLR13. We know that there are ribosomes in prokaryotes and eukaryotes differ, and you can clearly see here the prokaryotes have 23 sRNA, okay, as a ribosome subunit, whereas eukaryotes have 28 sRNA, and the sequence of 23 sRNA is unique one, which is recognized by TLR13. You can see here the sequence C double G triple A GACC is recognized by TLR13, and it can send signal downstream through MYD88. And so it so happens that this site is also important site for antibiotics to work. Okay, the antibiotic which work on translation machinery, this site is very important. And if you have a mutation in 23S RNA or a mutation at this site, this bacteria becomes resistant to TLR13 response, okay, as well as to this antibiotic group of antibiotics. Okay, now this was about the TLR signaling. Now we go on to the next class of pattern recognition receptors, where they found that even TLR3 null cells could produce interferon in response to poly-IC. Poly-IC mimics double-stranded RNA, which suggested that there exists another pattern recognition receptor which is independent of TLR3. While studying, they came across that molecule is RIG1, which is retinoic acid inducible gene family, which is RIG1 family. And there are three members in this family: RIG1, MDA5, and LGP2, which has an RNA helicase domains, repression domain, and a CARD domain. CARD domain is caspase activation recruitment domain. Here, CARD is caspase recruitment domain. MDA5 is a melanoma differentiation associated gene 5, and LGP2 is a laboratory of genetics and physiology too. So these molecules were identified earlier itself, but their significance was appreciated later when people started studying innate immune responses. Okay. So here, generally this helicase domain is repressed, but when it binds to double-stranded RNA, okay, here it gets released. Now the CARD domain here is free to interact with another protein. And this CARD domain interacts with a molecule here, which is called IPS1, and this molecule was identified simultaneously by 4 different laboratories when published at the same time. You can see here, and they called it differently based on their identification or how they derived this molecule. So IPS1 was identified in Dr. Shizuo Akira's lab, which is known as interferon promoter stimulator 1 protein. And till date, whenever I have looked at promoter assays, I have never found any potent molecule better than IPS1 to induce type 1 interferon. Okay. Then MAUS1, it's the same molecule, but they were identified as a mitochondrial antiviral signaling protein. VISA, which is virus-induced signaling adapter, or CARDIF, which is a card domain containing interferon inducing protein. Okay, the four groups identified same protein and published in around a similar time. And this IPS1 connects the innate immunity to mitochondria, because the IPS1 is anchored onto mitochondrial membrane here. Okay, and the pathway remains the same. The TBK1 has to be activated, which goes and phosphorylate IKK, and then there is a interferon gene activation. Similarly, as we can see here, that there is a specificity difference in MDA5 and that of RIG1. Okay, RIG1 identifies 5' triphosphate, which is not capped, and MDA5 identifies double-stranded RNA, which is supposed to be shorter than 200 base pair and should have palindromic sequences. Okay. Now that was derived by studying of different RIG1 knockout and MDA5 knockout fibroblast when they infected with a single-stranded RNA viruses, and it did not induce any interferon here, but MDA5 null cells could make it. But when a double-stranded RNA virus was infected to these cells, you can see that RIG1 knockout cells could make interferon, but not the MDA5. So it was identified that 5' phosphate group in RNA activates RIG1 and double-stranded RNA activates MDA5. That brings me to the third pattern recognition receptor class, that is a NLRs, or node binding oligomerization domain like receptors. Okay, there are many have been classified into this group because they are nucleotide oligomerization domain, and many of these families have nucleotide domain oligomerization domain within their structure. It is also known as NBS LRR family of proteins, which is nucleotide binding site or leucine-rich repeat elements, as you can see here, or it is also called caterpillar family, which is a card domain transcription enhancer, purine binding, pyrene domain containing, and lots of leucine-rich repeat containing molecules. They all represent the same family, but named differently as per their discovery. Now, it brings to the nodes. Nodes, when they were identified, previously it was thought that it is going to be LPS that is going to be the ligand for node 1 and node 2. But later on, when the purified LPS was employed to the experiments, it came out that it's a peptidoglycan that is a ligand for node 1 and node 2. And here I bring the differences in gram-positive and gram-negative bacteria. As we know, gram-negative bacteria has two lipid layer, outer membrane and a inner membrane, and has around only 20% of peptidoglycan layer here, whereas gram-positive bacteria has 80% of peptidoglycan layer and a plasma membrane here. Okay, and even the linkages between the peptidoglycan is different in gram-positive and gram-negative bacteria, gram-negative and gram-positive bacteria, and that is what is identified by node receptors. You can see here the gram-negative recognizes different linkages, and node 1 recognizes linkages in gram-negative bacteria, and node 2 recognizes linkages in gram-positive bacteria. So they can even distinguish between gram-positive and gram-negative peptidoglycan. Now, what is the significance of NLRs or node-like receptors? They are cytoplasmic receptors, as I said earlier itself. They are classified as cytoplasmic receptors. So in a gut where you are constantly exposed to the bacteria, the TLRs generally gets tolerized. Okay, only when the bacteria enters the host cell, that is where they encounter node-like receptors, and they come in contact with node-like receptors, and then they start producing cytokines, which will activate immune system per se. And this activation is important because it is alarming the system that some bacteria or pathogenic bacteria has entered within the cell, and this is an alarming signal. Now that brings to the NALPs and the concept of inflammasome. And before going to inflammasome, Now, what is IL-1 beta? Because the sole function of inflammasome is to catalyze IL-1 beta or convert pro-IL-1 beta into active IL-1 beta. Okay, so IL-1 beta is a very important cytokine in the immune system. It is implicated in various diseases like rheumatoid arthritis, IBD, MS, and osteoarthritis. It can be released by many cell types, and it has a very important role in septic shock, which is the most common death in ICU. And hence, the role of IL-1 beta in management of pain and inflammation is of intense interest. And with that, Duke Shop's lab followed the story to identify which is the complex that activates caspase 1, because we knew caspase 1 is the one that cleaves pro IL-1 beta into active IL-1 beta, which get released outside the cell. And when they, by immunoprecipitation, precipitated this inflammasome complex, the word inflammasome was coined by Dr. Duke Shop, and we identified this complex had caspase 1, caspase 5, NALP, and ASC, which is apoptosis associated spike-like protein here, which binds to this NALP and the caspase here. Okay. Generally, it is inhibited here, but moment it nitro domain interacts with the ligand, it opens up, and now the pyrene domain is open to interact with the card caspase 1 card domain, and then send the signal downstream, and the nitro domain causes oligomerization, by which the many NALP molecules can come together and send active signal downstream to activate the mechanism to activate IL-1 beta. Now, since I mentioned that IL-1 beta is a very important molecule, there has to be multiple checks and controls to control IL-1 beta and IL-18, which is a pro-inflammatory cytokines. So here is one first signal comes from TLRs, which leads to activation of NF-kappa B. There's a transcription of IL-1 beta membrane, and there's a pro-IL-1 beta which is formed. But it also requires a secondary stimulation, which is activation of P2X7 receptor, which is purinergic receptor, which senses ATP outside. So it says that not only TLR activation, but also there has to be ATP, suggesting high concentration of ATP outside the cell, suggesting that there has to be substantial damage outside the cell to release ATP, which may be required to fully activate this caspase complex. And only then the IL-1 beta is going to be released. Till then, it will not be released in amount that is required to alarm the immune system. So there are two signals which is known. Signal 1 and signal 2 is required to activate caspase molecules or caspase inflammasome complex. Now, so I would like to introduce a concept of DAMS, or danger associated molecular pattern. This concept was coined by Dr. Polly Matzinger in 1994, suggesting that when you have a dying cells or when you have a cell lysates, when you expose this cell lysates to immune system or dendritic cell, the dendritic cell gets activated, suggesting that there are molecules which are released upon the cell lysis, which will activate the immune system. And most prominent of them is crystalline monosodium urate. Now, what is monosodium urate? Is a uric acid, is present in cell, and it's a degradation product of purines. But under stress condition, there is a lot of uric acid that is being released, and at times it develops a crystals, okay, which is called monosodium urate crystals, and which is recognized as etiological agent of gout, which you can see here, swelling that is seen at the joints. And this gout is a result of NALP activation, okay, and this is triggered mostly by IL-1 beta, as I said. But if you put soluble IL-1 receptor, you can manage this pain and these symptoms, suggesting that IL-1 beta plays very important role, and NALP inflammasome also plays very important role here. Now, here the NALP3 activation is different than that of NALP1, where NALP3 can directly interact with various gout crystals, bacterial RNA, toxins, or many other products of the pathogen, and can get activated and can cleave IL-1 beta and IL-18 to make a active form of IL-1 beta and IL-18. Now, this is about NALP3. But then while studying this, they came to know that there is no requirement of NALP activation required for activation of inflammasome complex or IL-1 beta release. And that is where it was identified that caspase 11 can directly detect LPS and can get activated. So caspase 11 can detect LPS and can be causing inflammasome or inflammatory release. Okay, and there were two canon identification of inflammasome. One is canonical inflammasome, which is mediated through NALP1 and NALP3, and or there's a non-canonical inflammasome like caspase-11, which can directly recognize LPS and can cause pyroptosis. Pyroptosis. This is a new word because people have heard of necrosis, people have heard of apoptosis, but pyroptosis is a rapid cell death. Okay, around 80 to 90% of cells are dead within 10 minutes of exposure of this pathogen, and that is mainly caused. There are many molecules were put forth previously, but latest study shows it's mainly due to gasdermin D. Gasdermin D is a molecule that assembles itself on the cell membrane and causes a pores in the cell membrane that makes the cell swell and lyse. Okay, and this gasdermin D is also a target of caspases. Okay, so that's the reason how the pyroptosis gets activated, and pyroptosis really leads to rapid cell death. Now, that was about NALPs. We have seen TLRs, we have seen RIG1 family, we have seen NALP. Now we come back to DNA sensing mechanism. So way back in 2007 itself, when we were looking at various adapter molecules, Dr. Taniguchi's lab in Tokyo University had identified a dye, which is a Z DNA binding sensor, which was in cytoplasm. DNA is generally supposed to be in nucleus, but here if the DNA comes in cytoplasm, there are many DNA sensors in the cytoplasm which will bind to this DNA and can activate this immune cascade like TBK1, IRF3, and then it will lead to production of interferons. And there are many DNA sensing mechanisms that we know today, like AIM is one of them.
Dr. Taylor?
Yes.
Yeah,
Yes.
yeah. We're just running short of time here.
Sure,
Can you
sure,
please
sure.
wind up?
Yeah,
We are not left
yeah.
any time for any question, though we have one
Oh,
very good question here coming up.
okay, sure, sure, sure. Okay,
Okay.
so then, this is the last one that I'm mentioning here,
Hmm.
that which is a C-gas sting pathway, where there is a cyclic GMP-MP synthase, which recognizes again a single-stranded DNA or click DNA and forms this cyclic molecule, which is CGAMP, which is recognized by STING, which is a stimulator of interferon genes, which resides on ER. Earlier we had seen IPS1, now we see STING, which is in ER, and again it can release interferons. And lastly, C-type lectins, which not much work was done till time because most of the attention was taken by TLRs and inflammasome. But latest, I think last 4 or 5 years, there's a lot of attention on C-type lectin, which recognizes the carbohydrate molecules. And they do not have much lot of domains like TIR, but it can also activate respiratory burst and phagocytosis to kill the intracellular pathogens. So here I will stop, and at the end, I will just like to make a statement that we learned that evolutionary innate immune system have developed non-clonal receptors that recognize the patterns that are non-self, and there is a certain specificity to the innate immune system. So pattern recognition receptor, the sole purpose is to show that how a different pattern recognition receptor recognize patterns which is so much diverse and which is can distinguish between self.
Hi, hi,
Hi, Dr.
hi,
Preeti,
Madhurika,
Dr.
hi.
Arora, and everybody. Good afternoon. Yeah, I am here. Just starting.
Please get going. Video is
Good
on.
afternoon. Video. Why it is not showing? Start video.
Yeah, there you are.
Yeah. Yeah.
We can see you. Okay, go ahead.
So good afternoon, everybody. Today I will be talking about neutrophils. Though my research field is dendritic cell, macrophage, monocytes, but the requirement for this virtual pool is I should talk about something about the neutrophil and the neutrophil associated immune response, that is the innate immune response. So, if we start about the innate immune system, all the immunologists know, but as there are some students also, so I will be I have arranged my lecture like a popular lecture sort of thing, no data, nothing is there. So, regarding immune system, the activation of the complement cascades, recruiting
Hello.
the immune cells. Hello,
Hello.
hello.
Yes, yes, please tell me.
Yeah. Recruiting the immune cells to the site of infection,
Okay.
phagocytosis of
Please
foreign
tell me, this
antigen
mic
and
will drop. But
And
let me check if I can come today evening if possible to meet you. What is,
Dr.
yeah, yeah.
Yeah, Dr. Prafulla, could you please mute your mic?
and
Yeah.
the presentation of antigen class 2. And the last one is the direct clearance of the pathogen by generating reactive oxygen species and antimicrobial peptides, including the calcitonin and defensin. So these are the participating cells in the innate immune system, starting from the mast cell or the basophil, then the macrophages, natural killer cells, dendritic cells, monocytes, neutrophil, basophil, and eosinophil. So the last three panels are the granulocyte populations of the cells, leukocytes, and the main focus of today's talk is the neutrophil and the neutrophil mediation in immune response. So if I go quickly about the basic background of the neutrophil, what are they? They are the inflammation may be acute type, any type of inflammation. It is the first line defense the body just triggers when some pathogenic attack happens. And in case of this inflammation, there may be two types of inflammation. One is acute, which is short term, and another is the long term one, is the chronic one. And the hallmark of the localized inflammation is the vasodilation, means the blood capillaries, they just have enlarged volume of blood influx at the site of injury or infection, and the vascular permeability is also increased, leading to the leakage of the fluid from the blood vessels to the site of infection, causing some edema. And within few hours, the leukocytes also enter the tissue from the local blood vessels. And these entering and the interactions of the local cells and the pathogens are mediated by some receptor ligand mediations. Those are the PAMs and the DAMs, means the pathogen associated molecular patterns and the damage associated molecular patterns. And these are the recognition pattern-like receptors, which help to establish the connection of the cells recruited there at the site of infection or injury and the pathogen. Then the activated phagocytosis mediated by macrophages or neutrophils and the natural killer cells with the pathogens, they are the first response producing the additional mediators. And what are the additional mediators? They will be activated by the systemic acute phase and additional physiological responses, including the initiation, healing, and mediating the adaptive immune response. So the immune response are the starting point, and the adaptive immune response will be taking the baton from the innate immune response. Thus, the immunity of the body just get triggered, and the whole cell population, they just take part in a orchestral manner. And here I will be trying to play one video. I don't know whether it will be, no, I will be going to that video. Just wait for a while.
Can we just move ahead? Yeah.
No, sorry, it cannot be played. So we will be moving ahead.
So
Yeah, I am just moving a bit.
yeah, yeah.
Yeah, so
Thank
it's
you.
not showing. Yeah,
Start from this slide again.
yeah. So the mechanism of action of the local inflammatory response, it starts with the local infection, tissue damage, or exposure to some harmful substances, and the PANs, DANs, and the other crystals, they are the recognition receptor, and some receptors are um, expressed over the leukocytes called the neutrophils, macrophages, basophils, or the recruited leukocytes nearby the infection or tissue injury site, and the counter ligand is receptor over, expressed over the endothelial epithelial layer, and thus the recognition happens and the inflammatory response starts. In this inflammatory response, the initial components of cellular innate immune responses, these include the pro-inflammatory cytokines called the TNF alpha, IL-1, IL-6, other chemokines, prostaglandins, and the histamine. These are the other mediators which just take part to trigger the cells associated in these uh, cellular recognition. Now, if we go to this picture, it can be seen there are numbers given here 1, 2, 3, and 4. So, from the 1, if we start there, it is the tissue site which is being damaged. First of all, we have to just remember the inflammation may be caused by tissue injury or some pathogenic infection. Here we are taking the example of the tissue injury. If the tissue is injured from the outside, it is the epithelial layer of the body. Outside from the environment, pathogen invades inside our body system, just influxing the epithelial layer of the skin. And the tissue damage and bacteria cause these sentinel cells to come over here. And if the bacteria or any sort of pathogen is intruding inside the body system, the permeability of the capillary system, this is the blood vessel, the permeability allows an influx of fluid and the cell at the site of the infection or the tissue damage. Means there are the vasodilation occurs, the permeability of the blood vessels increases, and the fluid, they are allowed to influx from the capillary to the site of the infection, causing some edema or fluid accumulation, and the cells which are flowing inside the capillary, they are extravasated at the site of the injury. And the neutrophil, here we can see it is the neutrophil, the multilobular neutrophil having some granules inside this one. These neutrophil and the other phagocytes, it is the macrophage, they migrate at the site of the inflammation, and the phagocytes and the antibacterial substances destroy the bacteria. So these pathogens can be degraded directly by phagocytosis or just degranulating through these granules by the mast cells or the neutrophils or the other recruited cells. Now, these neutrophils, they not only engulf the bacteria, but also they have some other machineries by which they just entrap the foreign pathogens. That is, they release some granules and the formation of the neutrophil extracellular trap or net. If we see this picture, the net is looking like this. It is the net The mode of action of the neutrophils are: one is degranulations. The granules present inside the neutrophils are the proteases, phospholipases, elastases, collagenases, and these are the enzymatic granules which, when the neutrophil occurs degranulations, they release these granules, and directly it is the pathogen, and directly it can degrade the pathogen. It is one mode of action of the neutrophil action. Another one, these multilobular neutrophils, they can directly phagocytose the foreign pathogen, and this is the phagosome. Inside the phagosome, the lysosome, when it fuses with the phagosome, it becomes the phagolysosome. Inside this phagolysosomic vacuole, the enzymatic activity acts on the pathogens and they get degraded. Other than this one, another mode of action to entrap the foreign pathogen is the netosis, that is the neutrophil extracellular trap. It occurs by the neutrophil, and it is nothing but the histone and DNA coupled network sort of structure which feels like a net where mesh sort of a trap, and within this trap outside the neutrophil, if the pathogen is larger in size, which is difficult to phagocytose, then neutrophil takes this mode of action of entrapping the pathogen, and this is called the netosis. And these net entrap the pathogen, and through these myeloperoxidase, elastase, these enzyme-mediated degradation of the pathogen occurs. So, if we go through the mode of action of the neutrophil mediated foreign pathogen degradation, the another one we have to go through that the pattern recognition molecules, the receptors, the ligands, which helps to establish the neutrophil over the endothelial system more profoundly, because when the blood flows, the cells are very easily flown over the endothelial epithelial layers. So, to establish the full hold of the neutrophil, the pattern recognition molecules, they play an important part, and these are the pathogen recognition receptors, the C-type lectin receptors, toll-like receptors, nucleotide oligomerization domain node-like receptors. These are displayed over the neutrophils, and they help to interact with these ligands which are displayed over the endothelial venule, epithelial membranes. And if we see dendritic cells, natural killer cells, they are also taking part in the innate immune response triggering. And if you see these are the TLR receptors, these are the node-like receptors displayed over the dendritic cells. These are the C-type lectin receptors, the dectin, this and which are displayed over the neutrophils. And these receptors have specific ligands over the endothelial venules and the epithelial membranes, and by the establishment of a firm uh, holding over the endothelial system with the neutrophil and all these recruited cells, they help the host immune defense cellular functioning very well through phagocytosis, autophagy, apoptosis, and inflammations. These are the mode of actions by which the foreign pathogens are cleared. And subsequently, if we go more intricately, you can see if the bacteria Here the pathogen model we are taking as the bacteria. It can be any pathogen. When it invades the macrophages, they release the cytokines and the chemokines, and these cytokines and the chemokines, they just trigger the vasodilation and increase vascular permeability, cause the redness, heat and swelling. Whenever some infection, some allergic infection happens, the first line physiological change, we just see that it has some red rashes over our skin, and we feel there are hot flashes on the surface of the infections, or in case of systemic infection, the whole body, the fever occurs, and the edema or the swelling, it occurs. Now the inflammatory cells, they migrate into the tissue. Here it is the tissue, it is the capillary. So the inflammatory cells, they have to migrate at the site of the inflammation and the mediator that cause the pain. Now, the neutrophil, which is the specific functions of the neutrophil in case of immune triggering, we will be just going through that one, that it releases the granules generally, include various chemokines and the pro-inflammatory cytokines that includes the defensins, serine proteases, lysozymes, cathepsin, etc. And the defensin can act as the chemotactic factors for the other T cells, because we are talking about the monocytes and the granulocytes. We are not talking about the lymphocytes, that the B lymphocytes and the T lymphocytes, because when the innate immune response is triggered, subsequently the acquired immunity, the adaptive immunity will also be triggered in subsequent stages. So the cells, the lymphocytes, means the T lymphocytes for the cell-mediated one, and the CD4 plus T lymphocytes and the B lymphocytes for the humoral immune response, they will also have to be coming at the site of the inflammation, and in the orchestral manner, everything will be taking part. So the chemotactic factors are also triggered and released by the neutrophils. It is the component of the innate immune system, and subsequently the ROS mediated, means NADPH oxidase mediated, the respiratory burst and the production of the reactive oxygen species and the nitrogen species. They are the mediators of the immune response for the neutrophil mediation. Now, how the neutrophil migrate at the site of the inflammation? We will be studying that one. The first step, if we study this one, it is the endothelium, the layer of the first line layer of the endothelium for the HEV, high endothelial venules, and these are the neutrophils we are studying about. So the first step is the rolling. Rolling means the inflamed endothelial cells expresses certain adhesion molecules named the selectin. So selectin is expressed over the endothelial molecule, which binds to its counterpart or the ligand, which is the mucin-like cell adhesion molecules, which is displayed over these neutrophils. So the selectin and mucin integration happens, and the adhesion molecules help the neutrophil to roll over, to just stay firmly, and rolling happens. Now, if the rolling starts, then the next step is activation. How it activates the chemoattractant release from itself as well as the nearby cells. That during rolling stage, the neutrophils come in contact with several chemoattractants. Mainly, it is the IL-8 and macrophage inflammatory protein like the MIP-1 beta. These help them to just get triggered to release the other proteases and the granules, phospholipases, elastases, and everything. Now, if the triggering is happened, we have started from here, we have reached here that the integrations of the receptor and the ligand, we have gone through the rolling and activation. Now, what is the arrest? Because they have to sit there very firmly. This stage is called the arrest, and arrest is caused by the signaling molecule cascade on, where through the chemoattractants, the integrins over the neutrophil surface, they just display some conformational changes, and these are associated with some immunoglobulin receptor like cell adhesion molecules or CAMs. It can be ICAM or VCAM. So this part of activation and arrest is initiated whenever the neutrophil arrest sits there very firmly. Then the next step of the trans-endothelial migration happens. What is the trans-endothelial migration? This is the trans-endothelial migration, means from outside the endothelial venules, it is the capillary, it is the blood vessels, and within from the blood vessels, the cells, mainly the neutrophils we are talking about now, they will be just migrating at the site of the tissue which is injured, and through that one, the pathogen is already embedded there. Now, the role of Now, the role of innate immune system in allergic response, how it happens. Because in case of innate immune system, the first line methodology is triggered by the skin, the cell barrier, and the response is first seen as the allergic response. Allergy caused by the B cell, if we go for that one, though they are not included in the cascade of the innate immune system, but also as they are taking part, we will be just touching about that one. It is the plasma B lymphocyte. Plasma B lymphocyte means when the B lymphocyte, resting B lymphocyte get attached with some pathogen, they become activated and the size become enlarged. They secrete antibody. Some antibody are free flowing in the blood. Some antibody becomes engraved over the blood B lymphocytes. It is the plasma B lymphocytes. Whenever they release the antibody, the pathogen activated and or the sensitized mast cell or the basophils which are already exposed with some pathogen, they display the receptors. The receptors, which receptors? They are the FC receptors. These antibody released from the B cells, their FC portions, they just get contact with the FC receptors of the mast cell, and thus what happens? The cross-linking of the antibody through the receptor, it happens. It is the protein antigen, the antibody against which it is generated. It is the antigen, the pathogenic antigen against which the antibody has already generated. It is prevailing in the circulation. And whenever these pathogenic antigens, they just are recognized by the FAB portions of the released antibodies, and the mast cells which display this antibody IgE over the receptor ligand mediations, the cross-linking of the antibody happens, and these cross-linkings are the first line cell signaling cascades which are on to just give the inflammatory mediator release or the degranulations of the mast cells, and these degranulations cause the hypersensitivity reactions or called the Anaphylaxis, it is the systemic hypersensitivity reaction in which the sudden drop in blood pressure, narrowing of airways, shortness of breath, skin rashes, these are seen. Often we can see the anaphylactic shock in case of venom, of the insect bite and the stings, it can happen. So though it is the part of the innate immune system caused by the neutrophil or the mast cells we are talking about, the B cells and the B cell mediated antibody cross-linking also take one major step to trigger this um, release of the granules or the granulation. Now, if we go for the neutrophil mediated disease resolve, because why neutrophil? We will study neutrophil so much. There are some infections, neutrophil can cause the degranulation, neutrophil can directly engulf the pathogen, neutrophil can cause the net-like mesh formation by which they can entrap the foreign pathogen. So in some disease setup, we will be studying how the innate immune response mediated by this neutrophil can be triggered. In case of TB infection, Mycobacterium tuberculosis, it is our major concern of our country. Not only that one, the other third world country, they faces this sort of infections, and how the innate immune system are getting activated when the first line infection happens. The phagocytosis or the oxidative burst occurs and the engulf microtubules, the Mycobacterium tuberculosis, they are engulfed with the lysosomic activation. The engulfed bacteria, they are neutralized by the oxidative burst, as well as in case of netosis, they are also entrapped and they are killed. Through these oxidative bursts, the neutrophil enzymes, they they cleave the bacterium and the apoptotic cells, the released apoptotic bodies, they are degranulated, and this apoptosis is the crucial step for resolution of the inflammation, which otherwise can cause the damage of the tissues as well as the other steps. If it is the total system by which the Mycobacterium tuberculosis bacteria is dealt inside the in vivo system. One is just without any participations of the cells. Here the human neutrophil peptides, enzymes, and the reactive oxygen species, they cleave the bacteria. Here the innate immune system, these left panel are the innate immune system taking part to resolve the bacteria, the mycobacterium, and here the adaptive immune system. And in case of innate immune system, you can see the dendritic cells, macrophages, and the neutrophils. It is the multilobular neutrophils. They trigger it, it releases IL-8 and the MIF-1 and MIF-1 alpha, these chemoattractants and macrophage triggering chemokines, they are released to trigger the macrophage, which instead phagocytose the mycobacterium, as well as dendritic cell is triggered, which releases IL-12. And this IL-12 is the key cytokine which triggers the adaptive immune response, which is mainly through the T lymphocytes. And thus the pro-inflammatory response is triggered. Now, in case of viral infections also, neutrophil play a major part to resolve the viral infection. Here it is the neutrophil. We have already discussed the rolling, the activation, and the adhesion over the endothelial system. It occurs now. The extravasation means the transmembrane migration from the capillary to the site of the infection or the injury happens whenever neutrophil just influx at the site of injury. They take part in the similar way. They releases the granules or chemoattractants, which helps the macrophages and other cell types to take part in adaptive immunity as well as in the innate immunity. And in case of direct viral components which are free moving in the circulation, they can be entrapped by the net-like structure caused by the neutrophil. Here you can see here is the net-like structure, the DNA molecule these coupled with the histones, they have formed a mesh-like structure so that they can just entrap the pathogen. Here it is the viral component. So in this case, the viral infection is also displayed as well as dealt with the neutrophil surface protein called the CD43, CD45. These are the initiating molecules, initiating proteins, which in turn helps the total neutrophil population to get triggered and take part in the other one. Now, in case of autoimmune diseases, what are the autoimmune diseases? Till this point, we have read that neutrophil has triggered the innate immune system, and how the other cells, the chemicals, chemokines, cytokines, chemoattractants, and the cell adhesion molecules, which are taking part to trigger the immune system. Now, in case of autoimmune diseases, what happens? The neutrophil extracellular tract, the nets, they are the main culprit here because they are the DNA molecules. But what happens in case of autoimmune diseases? Antibodies, self-antibodies against these net-causing DNAs, they are formed inside the body. And when these self-harming antibodies which just recognize the net DNA component as the foreign component, what happens? They will be just degrading or just dissociating the net formation, and this type of self-harming behavior of this situation of the body, it causes some autoimmune diseases. And in case of systemic lupus erythematosus or the SLE, it is also having the same problem where the net is the culprit, the DNA component of the neutrophil extracellular trap is the main culprit. And other than this one, in case of the rheumatoid arthritis, if you go, it is the SLE problem, if it is the physiological um display we can see in case of systemic lupus erythematosus, when it displays this sort of rash and where the skin and the subsequent layers of the epithelial membrane, they become degraded and the skin becomes the most vulnerable sections of the body. And the main problem is the self-harming anti-DNA antibody formation. These antibodies, they directly kill, they directly degrade and recognizes as the pathogen one, as the foreign component one, the net antibody, net DNA antibody. Now, in case of neutrophils in rheumatoid arthritis, what happens? In case of rheumatoid arthritis, it is also an autoimmune disease where the cells, the inside antibodies, they are the self-harming one, which where the in-house proteins, they just trigger to build up autoantibody, and these autoantibodies, they are deposited in the juncture of our joints, and these joints become stiff, and the flexibility of the joints, the body system loses. And this is also a major problem where the neutrophil also take part. Here you can see the extravasation of the neutrophil occurs, and it recognizes these antibodies, and this neutrophil mediated antibody recognition happens, and the deposition here happens, which reduces the mobility of the joint.
Uh, I'll interrupt you, Dr. Manideepa.
It
Can
is,
we quickly wind up so we have
yeah, it is the last,
a
it
few
is the last one.
questions?
It is
Yeah.
the last one, yeah.
Okay, thank you.
So I will be concluding this one, the tumor-associated antigens and the in case of tumor, the neutrophil in the human tumor microenvironment of lungs, here the neutrophil acts as the pro-inflammatory cytokine releasing one, and in case of colorectal cancer, they trigger in the anti-inflammatory process where they just reduces the inflammation. And if we sum up the whole one, the anti-inflammatory agents that reduce the leukocyte extravasation, there are someone we use the steroids, and other than the steroids, the aspirin or the painkillers we use to reduce the inflammation, because whenever we feel some pain, because all the innate immune system, they trigger the innate immune system, the first line pathophysiology is the rash, infection, and the pain. So painkillers, the aspirins or NSAIDs are the reducing one, the leukocyte extravasation. And the one we have to just go touch the last one is if the extravasation of the neutrophil um, is hampered, what happens? It gives some problem, and it is called the disease setup called the leukocyte adhesion deficiency or the LAD associated complications, which include the defective neutrophil mobility at the site of infection or the inflammation. So, concluding one, our main first line defense mechanism to have the innate immune system, the neutrophils take birth, and within 2-3 hours of the infection, it just resides, it just migrates at the site of the infection, it takes charge, it releases the chemoattractants and degranulating chemokines and enzymes which cleaves, which degrades the pathogen. It creates some net-like structures which entrap the foreign pathogen, as well as it creates some, whenever it is the acute one, sometimes the acute inflammation extends to the chronic inflammation where the sepsis, in case of respiration problem happens. So,
Dr. Manideepa,
yeah,
Dr.
yeah,
Thank you.
yeah,
Thank
yeah,
you so much.
yeah,
We can have a few questions,
yeah, yeah.
because I can see a few questions being prompted on the question and answer
Okay,
bit.
okay,
And
okay.
if you could, let's start by asking the panelists if they have any questions. Then we go on to the questions from the participants.
Should I open the question box?
Yeah, yeah. You please open the question box. I can
Yeah,
read
yeah.
it out to you as well. Okay, we can start with the question. There is a question there from Michael Dureraj. He's asking that the multi-lobe nature of neutrophils, is there a reason why it is multi-lobed?
No, there is no reason, but it is the nature selection that all the granulocytes, they are the multilobular, dilobular, because the neutrophil, eosinophil, basophils, they have more lobes, and I don't think there is some specific reasons, but it is a natural selection where the granulocytes dispose this sort of multilobular nuclei.
Okay, so there is a compliment which says very
Yeah, thank
interesting talk,
you.
madam, from Anand Maurya.
Thank you. Thank
So
you.
we do the panelists have any questions? Any panelists particularly have?
I forgot to thank because I didn't have initially. So I want to thank Dr. Dean Afroz for this introductory talk about me, as well as to just compile all the sessions, and Dr. Arora and his associates and all our panelists as well as our participants. And yeah,
So
may I read? Yeah,
we have
one
little
question. Yeah,
time. We can just answer. This is a good question coming up, which
yeah.
says that neutrophil is working against Mycobacterium tuberculosis infection by a wide range of mechanisms. I want to know the mechanism in leprosy also. Is the same mechanism as that of tuberculosis, or is it depending on the spectrum depends on the immunological response to the patient of the patients? Did you get the question, ma'am?
1 second. I want to know the mechanism of leprosy.
What is the role of neutrophils in leprosy patients?
Yeah, all the
Anything?
time. All the time. What happens? Any infection, we went through all the mechanism of actions. When whatever it is, it may be the bacteria, it may be the virus, or any fungal infection. Infection is resolved by the neutrophil in three mode of action: directly by the degranulations of its lytic enzymes, that the peptidases and the phospholipase, proteases, and all these things. Number two is phagocytosis, directly engulfing that one. Number three is the network where the infection or causing pathogen is larger in size. They just create a net-like structure outside their cytoplasmic barrier, and they just entrap that one. So it is the mode of action in three ways they work. Not only that one, their chemotactic degranulation, they cause the recruitment of the other cells of the innate immune system called the basophil, macrophages, as well as the dendritic cells. They are recruited there and they take part. As it can be applied in every infection disease setup, whether it is leprosy, whether it is the mycobacterium or any viral component.
I don't see any more questions. So thank you, Dr. Manidipa. Thank
Thank
you,
you.
Prafula.
I need to say something.
Yeah, sure.
Okay,
Okay,
sir.
thank you very much. There is one
Thank you.
question in the chat box actually. What somebody has asked Dr. Sarah Dalai. He says, what would be the fate of immune response if neutrophils act as antigen-presenting cells?
I couldn't get, sir. Sir, please repeat.
What would be the fate of immune response if neutrophils act as antigen-presenting cells? If
Neutrophil act as an antigen presenting cell.
yeah.
Yeah, so in every setup, it is the same one. Antigen presenting cell will be displaying some antigenic component or epitope, and then the ligand will be displayed over its counterpart called the T cell, either T helper cell or the T cytotoxic T cell. And if the antigen is presented to the T helper cell, then it will be the humoral immune response triggering, and if it is the class 1 mediated antigen presentation to the cytotoxic T cell, it will be the cell mediated immune response.
No, that's okay. But I think Dr. Sarath has tried to ask if do the neutrophils they really act as antigen presenting cells
Whether neutrophil are presenting antigen.
different
Okay,
from cell or
no,
macrophages?
I don't think so. They don't take part like the antigen presenting cells. They just interact with the antigen, and they itself degranule and all this direct killing mechanism. They just take part. They
All
don't
right. Thank you
present.
very much. Thank you.
Yeah.
Thank you, Manipa.
Thank you, sir. Thank you, sir.
So, Dr. Dilafroz, you can wind up this session, please.
Yeah.
May I add a point, Dil, before you
Yeah.
conclude?
Sure.
Yeah. So just in continuation to Dr. Monidipa's response, I would like to add that neutrophils are very, very short-lived cells. Reason
Yeah.
being that the potent degranulation, which leads to release of potent enzymes, is very, very toxic to the cells. So
Yeah.
if they are antigen-presenting cells, then they cannot. These short-lived cells cannot be very effective antigen-presenting cells. That could be one of the reasons.
Yeah. Maybe.
Yeah, very
Because
well.
their main mode of action is degranulations of the lytic enzyme. The main.
Yes.
That
Thank
is
you.
why the granulocyte populations are popular to just trigger the immediate effect of the innate immune system. Otherwise, presenting antigen and just triggering the immune system, it will take some time. So innate immune system don't have that much of time. So they are very short-lived. They just get touched with the pathogen and they degranule, and by the one-to-one interaction, they just take part and just.
Thank you. We need
Sure.
to wind up.
Thank you, Dr. Monidipa. So with that, we come to the end